So I'm Summer Rosnowski, and today we're going to be exploring how to select and evaluate preprints for Journal Club using Europe PMC. During my talk, we're going to define what a preprint is, what a Journal Club is, and following that, I will introduce to you Europe PMC, and then we'll explore how to select and evaluate preprints for Journal Clubs using Europe PMC. So ASAP Bio provides a great definition for what a preprint is, describing a preprint as a scientific manuscript that is uploaded by the authors to the public server. It contains data and methods, but has not yet been accepted by a journal. A preprint is a manuscript shared before formal peer review, and it's open access, so it's freely available. Let's think about how the traditional journal peer-reviewed publishing works. You prepare a manuscript, which takes a few months, and you submit that to a journal. Then that's sent to peer review, and if accepted, the article is published within 4 to 12 months. So this process is quite time-consuming, and it may involve submitting to multiple journals and multiple rounds of reviewing and versioning the manuscript before publication. So that's before you can share those results. So this process does differ for preprints. You prepare your preprint manuscript, and you post that to the preprint server. Then your preprint is normally available publicly within 1 to 2 days. This, as you can tell, is considerably faster, meaning your research is shared with the community much quicker, and the community then have the option to leave feedback and reviews on your preprint. So then following potentially more data collection, new analysis, or following up with feedback and reviews from your community, you can update your preprint and release new versions. So this is called versioning, and these will go onto the preprint server, keeping the community up to date with the most recent developments of your work. So not only is the publishing process of preprints fast, but there's also other advantages to authors, including that preprints are given a unique identifier, a DOI, meaning they can be cited, and also many funders accept preprints as evidence for grant applications. Preprints are available alongside peer-reviewed articles in search tools such as Europe PMC, meaning you can search for articles and also preprints, so ones that are in peer-reviewed and also preprints all at once. And also most life science journals accept preprints, and many journals do operate a scoop protection policy that starts from the date of the posted preprint. Now we have an understanding of what a preprint is, we're going to explore what a preprint journal club is. Firstly, we need to determine what a journal club is. And it's a meeting where usually researchers gather to discuss the scientific literature. And these are usually articles in peer-reviewed journals. So evaluating preprint means reviews can be published. So this is why you may use a preprint over a journal that's been in the peer review process. And being involved in a preprint journal club allows researchers to keep up to date with current findings and develop essential critical thinking skills, as well as reviewing skills, improving your presentation and debate skills as well. So if you want, you can also share your feedback publicly, which can lead to collaborations and raise your profile as a researcher. As part of critically reviewing a preprint for a journal club, you will need to select and evaluate a preprint, which is suitable for your discussion. And this can be done using Europe PMC search and tools. Firstly, Europe PMC is a free global database of biomedical and life sciences literature. It's developed by EMBL-EBI and a partner of PubMed Central. It's a repository of choice for many life science funders, as shown here. Europe PMC contains a wide range of life science content, including articles, preprints, and other documents such as patents. This content is freely available as abstracts, full text, or links to free full text via the Unpaywall integration, where Unpaywall is a database that finds open access links for scientific literature. The way in which Europe PMC search works is unique because it searches through the abstract and the full text compared to other literature search engines, which search for keywords in either the abstract or the full text. Searching through the abstract and full text can help find more results, which is especially helpful for emerging research fields where fewer papers are published on the topic, and also potentially for preprints where you may have a lower number of preprints compared to publications in your field. And this approach also enables new search tools. For example, the ability to search specifically in article sections such as results and methods. And finally, it increases the visibility of the author's work, as there's a higher chance that their article will be found. Using Europe PMC to select preprints for a journal club is advantageous because it includes over 425,000 preprints from 24 different preprint servers, meaning you can search preprints from multiple servers through one search in Europe PMC rather than searching through individual preprint servers. So how are we going to select preprints in Europe PMC? One way in which we search for preprints on your topic of interest in Europe PMC is typing your search into the search bar here, and then selecting the preprints filter on the left-hand side under the search only type heading, which is shown here by the nice green tick. And you'll notice also that all the preprint articles have a little green box as shown on the slide, and this will always notify you that there's a preprint there, so that it's very clear that they're separate, they're different to the peer-reviewed journal articles. If you click to view a preprint, they will be viewed as shown here in Europe PMC. And the green box is still displayed here to highlight that it is a preprint. And you will also notice there's a yellow notice declaring that it may not have been peer-reviewed. Europe PMC also shows preprint version history. So you can be sure to view the most up-to-date version of a preprint and also be able to look back at previous versions if you wish. So that's shown in the green box. It will say V1, V2 to tell you which version and so on. And then you also have the versions down here so that you're able to follow the links and view whichever ones you wish. So this can be used to select the latest preprint version to review in your journal club so that you can provide the author with the most relevant feedback on their newest version if you choose so. In addition, there is also a notice on preprints that have been withdrawn by the authors, as shown here with a red box. This ensures that you can still see the information of a preprint, even though it's been withdrawn, but you're also aware that it has been withdrawn, which may influence your decision on selecting a preprint for your journal club. There also is a notice displayed to let you know if a preprint has been published in a peer-reviewed journal, as shown here with the red box going around that orange, and this allows you to link to the published paper. So there's various ways to search for preprints in Europe PMC, and one way to do so is by if there's a published version or not. So following typing your search terms into the search bar, you can select the preprints on the left-hand side under your search only filters, shown by the first green tick. And then you can limit that further to preprints that have been published in a peer-reviewed journal. And that will limit the search further. So as you can see for this search, there were 903 preprints found in total, and 413 have been published later on in a peer-reviewed published journal. So that's one way that you can use this to select which preprints you're looking for. Alternatively, if you want to search for preprints without journal published articles, if you write your search terms in the box, select the preprints filter again on the left-hand side, but then add the syntax, open bracket, all in capitals, has underscore published underscore version, :n, close brackets. This is telling you that you don't want a published version. And as you can see, this returns 489 results for the preprints without the published versions for the same search terms. Another way to search for preprints in Europe PMC is to search by author. So you can search for last name and first initial to find literature by a specific order, and then select preprints again so that you're getting preprints from that specific author. So as you can see in this case, for this search, there are two authors that come up with the last name Sander and initial M. So Europe PMC would return preprints for both of these authors. If you were interested in only one specific author, there is a way to differentiate further. So the first thing you want to search for the first author listed here, if you want to search for the first author listed here, you can do so by using their ORCID ID. Your ORCID ID is a unique identifier given to each individual researcher. So this ensures that even if authors have same last names and initials, you can still determine who wrote each preprint. So using that ORCID ID, I've determined which of these preprints was to which author. So you can use that. And the way that you would search for that, if you don't know the researcher's ORCID ID, you can view that in the researcher's profile and then retrieve their ORCID ID. Then to search for that in Europe PMC, you would do so following the syntax in the red box there, and you would type in their specific ORCID ID. So then that means that you will only see those articles from that specific author. Make sure you select preprints again so that you're only bringing those back. If for your selection criteria, your preprint is to be from a specific or a group of specific preprint servers, you can search by preprint server in Europe PMC. So under the usual search bar, there is an advanced search button that you can click on, which is shown in the red box here. And it takes you to the advanced search page. And then if you scroll down to bibliographic fields, and you can begin to type in a preprint journal, such as bioRxiv as shown here, then this will only return you results from either the one preprint server you've chosen or the multiple ones you've chosen to select. Alternatively, you can also search for preprints by changing the search order to times cited. This will provide you with preprints that were most cited at the top first, and then going down to least citations. Another way is also to use published reviews. So at the bottom of the search, again, we can go back into our advanced search. And once you go onto the advanced search page, if you scroll down to external links, if you click on the arrow, you'll get a huge box like this pop up. And then you can select each preprint review platform you'd like to include in the search to select for preprints with linked reviews. So they may be from journal clubs, they may be people that have decided to sit down and write them themselves. And if you decide to make yours publicly available, if they become published and are included in Europe PMC, this is where you'll find them. You can also set up email alerts, and this is a really good way to utilize selecting preprints for a journal club. So to do this, I would type in your search term or interest and select preprints on the left-hand side. And then I would go along and click this save and create alert. This will pop up a box that looks like so. And then you can give this a name, decide how often you would like it to come in and what day you would like to receive this. And these will come straight into your email inbox. So this will bring you new preprints that have been included so that you're keeping up to date, and then you can bring new ones to your journal club. Do bear in mind you'll need to log in for this because it will require your email. So following now that we know how to select preprints, how are we going to evaluate preprints in Europe PMC? One way to evaluate preprints in Europe PMC is via the sections. So today we're going to explore citations, impact, data, and reviews. Under the citations and impact section heading, it displays citations of preprints, which includes a citation graph. This graph monitors the number of citations year by year. So you can see for this preprint, it was cited 44 times in 2020. And then there's also links to the data records and reviews that cite this preprint, making it convenient for you to evaluate preprints by citation impact and to get a broader understanding of the literature field via the links to articles citing this preprint. You can also evaluate preprints impact by alternative metrics, including OUT metric and smart citations by site. Site powers smart citations, which provides context to the citation of the preprint, where the citation is classified to determine if the publication citing the preprint provides supporting or contrasting evidence for the cited claim. This information can be used to help evaluate the preprint. As part of your critical thinking, you're able to find evidence easily of supporting and contrasting data. OUT metric is a complementary metric to citation impact, and it assesses the impact and influence of the preprint. And the metric includes peer reviews, citation in public policy, blog mentions, mainstream media coverage, and mentions on social media networks such as Twitter. Another section is called the data section. So this includes data that cites the article and data behind the article. So the data sets in this section are identified through partner data resources and links to over 60 different life science databases, such as PDBE and EMBL, which can be used to evaluate the data in the preprint article. So data that cites the preprint contains links to databases where this preprint is cited. So this links the article to data accessions, which are unique identifiers for the data and the DOIs. This means where databases such as PDBE have referenced the preprint that will link to show up this section. And you can think of this as links to citations, but rather than to papers, it's to databases. For example, if a protein structural feature has been determined in this preprint, PDBE may use this information in their database on their page for that protein to now show that new structural feature information. And therefore, they will reference and cite this area, which you can then find here. So this then allows you to really help review and evaluate the preprint with ease, where the data behind the article links to the relevant data that is deposited in data resources, such as UniProt, which provide protein sequence and functional information for proteins. The data record in the corresponding database you are linked to existed before the preprint was published and is relevant to the preprint, but the preprint isn't cited there as they didn't contribute to the information on this database. You can think of these links as links to background information. For example, if the preprint refers to a protein, there may be a link for the protein to the UniProt base where you can find out the sequence of the protein and functional information, which is useful for background information and allows you to build a greater understanding of the content and research field related to this preprint to critically assess the research in the preprint as part of the evaluation process. Another Europe PMC tool that can support those running a preprint journal club is the review section. This provides links to reviews and recommendations from expert sources. This allows you to assess those evaluations, which can help you develop your own critical thinking and evaluation skills. They also provide an example of how to approach reviewing a preprint and even allow you to compare your own reviews to theirs. And don't forget that sharing your reviews increases the discovery of your preprint journal club and aids in the building of your profile in the community, research community. Another way to evaluate preprints in Europe PMC is by the annotations function, which can be found here on the right-hand side of the article, as shown in the red box. So in practice, when you open the annotations box, it pops up and you can select an annotation, which provides a list of biological concepts found in the text on the right-hand side, which is in the panel there. Those concepts include biological entities, relations, and interactions, for example, gene ontology terms, cell lines, or gene mutations. And they have been identified by a variety of expert text mining groups. There's over 40 different annotation types, including gene-disease relationships and experimental methods. This feature is a way to quickly scan a preprint to get an overview, find key concepts, and discover evidence which can be used in the critical thinking and reviewing process of a preprint as part of your journal club. So you can see here the individual terms are grouped by annotation category and the number of instances where the term was found, making it easy to navigate the annotations in this preprint article. For chemicals and proteins, a structural view is available, and all annotations are linked to the underlying record in a biological database, for example, UniProt, PDBE, and ENA, where you can explore the data in more detail to provide more background information to assist you in evaluating the preprint. To summarize, preprints are open access manuscripts that are made available before formal peer review. Journal clubs are meetings where scientific literature is discussed, and preprint journal clubs focus on discussing preprints, which build multiple essential skills for researchers, including critical thinking, reviewing, presentation, and debate skills. Europe PMC is a free global database of biomedical and life sciences literature. You can select preprints in Europe PMC via different searchings, advanced search, filters, and also setting up email alerts. And you can evaluate preprints using Europe PMC tools, including citations and impact, data, links, preprint reviews, and annotations. I'd like to thank you all for listening, and I will pass over to Esther for her part of the talk.
So, hello everyone. My name is Esther, and I will take you today through this preprint journal club that we run cross-institutionally through the University of Oxford, Mount Sinai in New York, and the Karolinska Institute in Stockholm. And we are hoping that this will bring many people into this adventure of preprint journal club, because actually we do this in immunology, but this can be extrapolated to any other field, really. So just that said, just a bit of introduction. So preprints appeared actually long time ago, and it was in the 60s that NIH launched this forgotten experiment, but then just to have like this fast exchange between scientists, but then this was completely abandoned after like the main journals opposed to it. And in the 80s, it was the physics and engineering fields that were using it more. And after the appearance of internet, there were the institutional repositories that were created. But there was a big change when bioRxiv was created in 2013. And then in life sciences, it started to be more and more used, especially the neuroscience field is the one that was making the most of it. And this actually changed drastically with the pandemic. When SARS-CoV-2 appeared, there was an explosion of preprints. And also here you can see this data for medRxiv. The downloads also increased a lot. So obviously these preprints have been very useful for how we have understood the pandemic, the immune response, the virus. And so there was a need to digest all this massive literature of preprints on COVID-19. And there were two independent initiatives that were done by all these researchers that wanted to help, and they were at home. And there was all this literature that needed to be filtered somehow to see what was relevant to the field. So there's these two initiatives, one in Mount Sinai in New York and the other at the University of Oxford. And what we basically did was that every week we would select the students and postdocs different preprint, and we will make a short summary. This will be uploaded to our website, and some of these summaries would be selected to be highlighted by Nature Reviews Immunology. And this was coordinated by Nicolas Vavret in New York and by Felix Richter in Oxford. And then we got actually some of these research highlights got very highly cited. And so from this, when things were opening up and going a bit more back to normal and people were going back to the lab, there was not this urgency of summarizing preprints every week. And so there was the idea of doing, summarizing a lot of preprints on COVID every week. But there was this idea of this cross-institutional journal club between Oxford and New York in Mount Sinai. In Oxford, it's the two different institutes. There's the Kennedy Institute and Oxford Immunology that actually was before viral immunology. And we were hunting for a third institution for a while. So we ran this for like 9 months. And then last November, it was the institute, the Karolinska Institute in Stockholm that joined this journal club. But why would you do a journal club on preprints? So basically, a journal club on preprints, you can give a very fast feedback on the authors. So these preprints are actually very close to be finalized. There's some studies that show that these preprints don't change much when they are published later on. It's also participating on this community review. It's going to be an open and transparent review of this unpublished work. This review also will focus on the science rather than the journal, like other reviews like Review Commons do, other initiatives. And also this would contribute also to some more healthy academia. So we would train these future team leaders to peer review on some unpublished work. So our main objectives are to train students and postdocs for peer review. These would develop their critical thinking. It will boost the discussion and interaction between students and postdocs. And we also provide a comprehensive review that can help these authors that are actually in a peer review process, because some of the preprints are already submitted to a journal and are undergoing to this peer review process. This kind of overlaps with one of other main objectives, which would be to build on this healthy peer review mindset, because we would deliver this independent, transparent, and more democratic. It's the students also that will evaluate the work. And also we have some interaction with the authors. And this also overlaps with our third objective, which would be networking. And other than interacting with the authors, we foster also the interactions between the students and the postdocs when we discuss, and also this would link with the faculty. I would explain this later. And also we create this community because we do this cross-institutionally of different institutions that work on the same field that might have different approaches, and that this can be two collaborations. So how do we work? So there's the preprint selection that Summer very nicely explained. And then there's one presentation per week. So we do once a week, a 1 hour presentation. There's 2 presenters that will do a 20-minute presentation with 10 minutes of discussion after. And then there's the evaluation, and I will go more in detail after. And after the evaluation, the presenter will write a preprint digest that will be uploaded into our website. And then through the evaluation of the journal club attendance, we will select 2 preprints every month that will be highlighted in Nature Reviews Immunology. And this will be the presenter together with a member of the faculty. And this could be a PI, but it could be also an assistant professor. In Oxford, we don't have assistant professors, so it could be a senior postdoc. So how do we select these preprints? The servers we usually use are the ones for our field, the more relevant for our field, so bioRxiv, medRxiv, Research Square. And here I'm showing you some data that we analyzed, that we don't really have a preference for specific journals. So this is about 50 preprints that we had selected and now are published. So you can see that it's quite spread. It is true that we selected more of some of the journals that are very related to our field, and that kind of makes sense. And actually, one of the ones that we made more highlights, actually 2 highlights, it's Nature Immunology, that obviously, because we do a preprint journal club on immunology, so this makes a lot of sense. So, but it's quite spread on all the journals. There's no like a preferential journal that we are selecting. And so for the presentation, how we do it, we alternate a presenter from each institution. So you can see down here, sorry, I thought I would have this, so I'm gonna put the highlighter, no. The laser pointer. So you can see that there's like someone from Karolinska will present, and then the second presenter would be Mount Sinai, and then Mount Sinai-Oxford, and the next week would be Oxford and Karolinska. And we do that by an Excel file that people would put their email, their affiliation, and then the select preprint. And what we analyze though from this is that when people present in the first slot, there is a bit of a spread when we score the preprint, and the second lot it's more concentrated. And this leads a bit to a bias on the presenter on the first lot having more probability probably to be selected for Nature Reviews Immunology. But we have to say that there's also been some presenters that presented in the second slot that didn't after wanted to highlight the preprint that was selected. So it might be also not that extreme. And how do we evaluate these preprints? So we evaluate these preprints by a Zoom poll. So we ask 4 different questions. Each question is 1 to 5 for each category. And we assess the quality of the preprint, the data that's been presented, but not the presentation. And we assess for novelty. So what's new from these findings? What is the scientific quality of the data? Is there the good controls? What's the significance for our field for this? And then lastly, we ask which institution are you coming from? And this we do it to know if there's a bias on the data that it's of this evaluation. And then, so when we choose the preprint for Nature Reviews Immunology, we choose, as I said, 2 preprints per month. We choose the best scoring, the one that scores the best for the 3 categories. So it will be out of 15, but we have a quality control of a minimum score of 11.5. So something that is below 11.5, we do not highlight it, even if we get the highest. That's what we do, up to 2 preprints per month. Some months we have done only 1 preprint. And so our system of evaluating preprints seems to work pretty well. So the ones that have been selected for Nature Reviews Immunology are the ones in red. So this would represent about 20% of the preprints that we present. There's no clear correlation between the score we get from these preprints and the impact factor these preprints are after published in the journal they are published after. But what we see is that when we check whether the presenter is from a certain affiliation, there's a bit of a bias on the score. So when someone from affiliation 1 would present, they would rank higher. Also for affiliation 2, it would be a bit higher also. But we discovered that when after the November, so when we introduced Karolinska into the journal club, this was kind of equilibrated. So introducing a third institution helps unbias the score we give to the preprints. And so when there's when the presenter finishes the presentation, they would upload this preprint digest that we uploaded in our website, and it will have this structure. So there's the keywords of the preprint, what's the main finding, what are the main limitations, and what is the significance and the novelty of this preprint, and then we will give credit to the person that did this review. And this is linked into bioRxiv. So into bioRxiv, you can go to this button that it's community reviews, and then it will bring you to read on Preprint Journal Club, and it will bring you to the page where we have this review on our website. And then we do the preprint watch at NRI. This is an editorial in February 2021 when they presented this initiative. And this is the tweet from the editor of Nature Reviews Immunology presenting this initiative. So what they highlighted was the value of the preprints, that they're rapid and open, that this the future of science, there's a lot of networking, as Summer also explained. You can claim primacy when you post a preprint, and also all the need for caution for these preprints because they're obviously not peer-reviewed. So when we highlight a preprint at NRI, we don't have a difference in impact factor from the ones we've chosen to highlight to the ones that have not been highlighted. However, when we stratify this, we can see that the ones that have been highlighted at NRI tend to be in higher impact journals, like there's very low highlighted preprints that are in low impact journal impact factor journals, and then I wanted to show you a bit how it works, our website. So we have a website that it's preprintclub.com, and we explain a bit who we are, what's our mission, what are our objectives. We also have, as I said before, the links to all the reviews of the preprints. We also highlight the ones that are after published in Nature Reviews Immunology. And also we have a page for the publication. So you can directly go to the ones that have been actually published. We also show who we are in coordinating this preprint initiative, also the participants, and I think this is important because it highlights a bit what everyone is doing. Actually, when people present their preprints in the beginning, they kind of explain a bit what they do. So this facilitates a bit of networking between the students that participate in this preprint journal club. And so we explain the profile, what do they do, and where do they work. And then we have a part of contact. So in any case, anyone wants to send a message, also this would facilitate contact with the authors. And we have some Twitter account, it's Preprint Club. And this has led to a lot of interaction with authors. So many different authors have interacted with us, especially to thank us and to be and looking forward to read what we wrote about their preprint. Helping, I think that helped a lot also in the perspective, because maybe with a peer review, you might get only one perspective. With this, you get different ones. And with this, that's it. I would like to thank the preprint, all the preprint club organizers, especially Felix, Nicola, the other Nicola and Laura from Karolinska, all the PhDs and postdocs that have been presenting and discussing these preprints every week, all the PIs and faculty that support this initiative, our institutions, and of course, Nature Reviews Immunology. And with this, I will take any questions. And I just would like to say that we are looking for people that are working on molecular cell biology, because we are looking for people to start the same, but in this other field. Thank you.
All right, so with that, we will move into questions. So we already have a few questions here. Oh, that's very good. So first question is from Gary, and it's for Summer. So what reviews are linked to a preprint on Europe PMC? Are they the reviews only that are annotated on Europe PMC, or are you able to pull across reviews from other reviewing platforms? For example, comments on bioRxiv, preprints, or the Preprint Journal Club community reviews?
Wonderful, great question, Gary. So for Europe PMC, you don't do the review itself through Europe PMC, so they are all pulled from other platforms such as bioRxiv and the Preprint Journal Club community, some of which were highlighted on one of my previous slides. So there's a few of those that they'll link to. So yeah, they're all pulled across and in. I hope that answers your question for you.
All right, thank you. Next question is for Esther from Gary again. You showed the tweets in response to your reviews. Do you have any other communication with preprint authors before or during or after the journal club process? Do you have a sense of how much of your feedback is incorporated into the preprint before or during a journal submission process?
Yes, so we don't communicate with the preprint authors before nor during the journal club. What we do is we announce which preprints we're going to present on Twitter, and we try to link it with the authors so they are aware of it. And this usually starts like some just tweets saying how they are looking forward to hear our feedback. But we don't communicate in any other way with them. What we are trying to do now is we want to know exactly this, how much of this feedback is incorporated, or if they are aware that there's this initiative, because obviously there's PIs that are very active on Twitter, but there's PIs that don't have Twitter, or researchers that don't have Twitter. So what we plan to do is to send the authors, after we've done the review, kind of a questionnaire, so we can understand if we are having some kind of impact and if they are aware that we are doing these reviews.
I think while we are waiting for other questions, Esther, if you don't mind, if I can ask a question. So researchers, I mean, while they submit their research to any preprint platform like bioRxiv or others, at the same time, I think they also submit their research to a scientific journal where they want their research to be published. So do you have any data, or if you heard from someone, that how much of these community reviews impact on the editorial process in the scientific journal? Or do the editors or reviewers there, do they consider community reviews as well?
I don't know the answer for that, but that would be really interesting, whether this influences the decision of the reviewers in the peer review process from the journals they have submitted. But no, I don't know this.
Is there any other general thought, Esther or Summer?
Yeah, I have a brief thought. As I just finished my PhD quite recently, and especially following Esther's talk, it's something I really wish that I'd been involved in, especially if you're kind of looking to go on to postdocs and progress in academia, because the networking that you can get, and with the Twitter as well, that's absolutely amazing, really important. I know so many people that have found their next positions through collaborations where they've met on Twitter or through sort of those sort of networking abilities. So that's amazing. And also it's the only chance that you kind of get at reviewing papers publicly until you're kind of in a much higher position. And that must be really kind of nerve-wracking if you hadn't had an opportunity to do it. So I think that's really great. It's an amazing scheme that they've organized there.
Yeah, if I can add, yeah, I think this helps raise your profile a lot, and many students are very afraid to present at the beginning because obviously there's people they don't know. It's 3 different institutions in different parts of the world, so sometimes they are a bit nervous about it. But I think if they continue coming every week, then the faces are quite familiar, and it's easy to reply to the questions. It's a very friendly environment, and I think that helps a lot on their presentation skills, and also if they get selected, for example, they get a publication out of it. So it's really good for raising the profile.
Thank you. Yeah, I think we have got 2 actually interesting questions. So do you also display your review on more platform than one?
So our review is in our website, sorry, and it's linked in bioRxiv that I'm aware of. I'm not sure if there is if there's a link in other preprint servers, but no, we display it there, we announce it on Twitter, and then if it gets selected, then it is published in Nature Reviews Immunology. But it's open to everyone from our website.
So the next question is about the journal club. So there will be a new journal club. Can you attend without being in the mentioned institutions?
Yeah, so if you're interested on attending our journal club, we have the contact part on our website, so you can contact us. And we have had people from other institutions joining. That's why in the evaluation we have other part when it's either Oxford-Karolinska and Mount Sinai. There's also others. So if people are interested, they can contact us.
All right. So the next question is for Esther. Do preprint club people register to the website? It's about preprint reviewer recruitment network, ASAP BIOS peer review networks, preprint peer review platforms, and journal might be very motivated to recruit great reviewers who engage in such journal clubs.
So we don't do as a preprint journal club. If people do it individually, they do, but we don't automatically, because people are doing the preprint journal club, register as reviewers in ASAP Bio.
All right. So next one is specific to COVID-19 preprints. Will the planned journal club in molecular cell biology be on COVID-19 preprints as well?
Yeah, sorry. So maybe I was not clear, but the preprint journal club we are doing now, it's not based on COVID-19. We might do some on COVID-19, but we don't do, most of them are just on immunology. So it's an immunology. It started with COVID-19, but then it's now preprints on immunology. So the journal club in molecular cell biology wouldn't be in COVID-19. It would be on the preprints that are recently uploaded. Usually we tend to present preprints that have not been there for long, so they are not published immediately. So we try to pick fresh preprints also.
Just out of curiosity, I have got another question here. So have you experienced like any comments from anyone who has submitted their data on a preprint website like bioRxiv? Something about where they're afraid of being scooped, because like in scientific publications, sometimes you don't want to share your data. You feel that it will be taken by someone else. So have you experienced such comments or discussions in your journal club?
Not in the journal club, but me personally, I know people that do completely the other way around, right? They want to show that they're the first ones to show this, so they put it in the preprint servers. Not to be scooped precisely, probably to show that they have been first on that. But we haven't discussed this in the journal club.
I'd just like to jump and add on that, if that's okay, because I've had quite a lot of discussions with quite a few different people that are involved in preprints. And one of the things that comes out is a lot of the time they're actually, they prevent you being scooped because you're showing that you've got this research already. So it might take another year or so before you get that publication out, but you've already got your preprint there. So it's already set. And then there's scoop protection from from preprint servers. So that's already set there as well. So I think in that sort of regard, that they can be seen more as a protection of scooping. And also, I think one reason scooping happens is researchers don't always know what other researchers in the field are doing because you've kept it quite quiet. So you've got something big, and then two groups are actually doing the same thing. And it's not always necessarily scooping on purpose. It's just you've got your research a bit quicker. But if everyone's got their preprints out, you know what everyone's up to. And if there's that transparency, then you're less likely to get those sort of competitions that you don't know you're part of.
Yeah, also, if you see that someone is publishing something very similar to you, you can take a different approach. So it might help not having very similar papers after.
Yeah, and you could even message them and be like, hey, we've been doing something similar, and then you can compare, and potentially that's a better paper and you've got more evidence and make a better story.